All right. Good afternoon, everyone. Thanks so much for joining. I'm Dr. Lauren Esposito. I'm the curator of Arachnology here at the California Academy of Sciences. And I've also had the distinctive honor of being the graduate student advisor for Kate Montana for the last 2 years. Today, Kate's going to be presenting her finishing talk for her master's entitled Uncovering Histories from Academy Scientists to Spider Evolution. If you have no idea what that means, I'm sure she's going to spend the next 40 minutes or so telling you. But before she starts, I just wanted to say a few words about Kate. I first met Kate in 2019 when she joined our Summer Systematics Institute, which is our undergraduate research program here at the Cal Academy. And she was not my student. In fact, she was a student studying sea slugs or nudibranchs. And so I never imagined that I would have been able to coerce Kate back in 2021 to study spiders, but somehow I did. But I think that is really exemplifies what is so exciting about Kate, which is that she's pretty much up for anything. I think if there's one thing I could say about Kate, it's exactly that. Like, Kate is always down to try just about anything that comes her way, as long as it seems intellectually stimulating or at least a little bit fun. Kate graduated from UC Berkeley in 2021. Is that right? 2020. Okay, 2020. I'm wrong. And I think one of the coolest things about her time at UC Berkeley that I learned about her after the fact was that she was certified in pyrotechnics while she was there. Which I think, again, comes back to Kate always being down to try just about anything. She's had a pretty remarkable time and journey here at the Academy. First of all, she has been a big part of the Academy community itself. She was a graduate student representative for our Curators Forum, which is no small feat to have a graduate student in a room full of curators once a month, listening to them hash it out and having the responsibility to report all that back to the graduate students. She also presented to our entire Academy community at an all-staff meeting on a really complex and difficult topic, presenting about the history of racism here at the institution, which you're going to hear more about in her talk today. But that's not all she did. During her time here, she mentored 14 students from high school through undergraduate. By the end of the summer, I'll go out on a limb and say, we'll have published 5 scientific articles ranging from archival research about humans here at the Academy, to nudibranchs, marine organisms, to spiders, to frogs and amphibians. And so I think that like, Kate really is all over the place. She's done just about everything, and I can't even imagine what she's going to go on to do next. But in addition to all of this, she also was the recipient for 2 years of a really prestigious scholarship from San Francisco State University, the ARCS Scholarship, which I think exemplifies really like the incredible tenacity and promise of Kate as a human. And she also participated in the Gold program at San Francisco State. So she's graduating not only with a degree in biodiversity science and biology, but also a certificate in data science. So I, for one, will be bummed to no longer have Kate as a student, but I'm hoping that I'll coerce her to continue staying here at the Academy for at least 1 more year. So with that, I'm going to turn it over to Kate. Thank you.
Great. Is this mic working okay before we start? Check, check. Can you hear? It's hard to hear. It's hard to hear. Is there anybody who can help with that? If not, I'll project. Great. Well, thank you so much, Lauren, for the beautiful introduction. And I am so honored today to be sharing my master's research with you. It means so much to see a room full of people that I care about and care about me. And hello to the people online. Thank you for tuning in. Let's get started. So, as one of the largest natural history museums in the world, the California Academy of Sciences, where we are, has one of the most complex archives and collections. These are two ways of documenting the history of organisms and the organization. I'm going to give you a few examples of how we have used the archives and the collections in the past. The archives have served as a historical record of both the institution of the Cal Academy as well as the city of San Francisco. They've also been used to put together a book about the famous Academy Galapagos expedition in the early 1900s. Our collections have been used to track things like disease transmissions in animals throughout time, review heavy metal deposition in those specimens throughout time, and keep a record of extinct species. For my research, I've used both collections and archives in novel ways, specifically to tell the untold histories of both people at the Academy and spiders. In the archives, I've been uncovering biographies of people who were not necessarily the most powerful or fairly compensated during the time that they were active. I use this project as a means of empowering and supporting the growth and development of emerging young scientists. Together, in putting together these biographies, we saw more of ourselves in the stories of people who are marginalized like us. And as for collections, I put together a genomics project, and many of the samples that are in our collections and the collections we borrowed from around the world were maybe not necessarily collected for genomics work. In all, my goal is to illuminate the dark corners of our knowledge and shine a light on groups of people or organisms whose stories have yet to be uncovered. So today I'm going to tell you some stories about working in the archives to empower emerging scientists, give you an overview of my spider phylogenomics project, and share my dreams and plans for the future. So let's start with discussing the archival project first. Our goal with this project was to empower young scientists by inviting them to collaborate on academy projects and to bring together existing internship programs across the academy in order to work on this collaborative venture. Our project was a pilot of a near-peer mentorship model, which involved 4 high school interns, an undergraduate student who co-mentored those interns with me, a graduate student, and also academy researchers. And together we developed a scaffolded 1-semester research experience for those high school students. The programs that this combined were the Careers in Science program. One, which is an internship that can run throughout the entirety of a local high schooler's time in high school. And interns from the Careers in Science program spend the academic year doing things like working on the public floor, talking to guests, all the way up to contributing to existing research projects here at the Academy. We also worked with the Summer Systematics Institute program, which is an NSF-funded research experience for undergraduates in which students from around the country come to the Academy for a summer to work on a research project. And I'm an alumnus of this program, and it's a big reason why I'm here right now talking to you all. So it's particularly special to me. And how we were going to achieve all of this was working on a research project in order to uncover stories of Academy scientists throughout using archival materials here at the Academy, and then collaboratively publish biographies on them. So let me introduce you to the team that worked on this project. We have Kareem Kaseta-Khoury, who is an undergraduate student who was with the Summer Systematics Institute. And together, he and I co-mentored 4 high school students, Raquel, Viva, Gabby, and Marion. And we also had, of course, the head librarian, Rebecca Kim, who provided us with much guidance and foundational support throughout the project and the mentorship process. So the project started with me doing a whole bunch of background work. So I sifted through dozens and dozens of archival materials here at the Cal Academy. This includes Academy annual reports, member newsletters, correspondences, and the like. And I found 36 candidates that we could look into further, and then we narrowed it down to 7. In January, when Kareem came on, together he and I put together a schedule for the rest of the semester and built up the curriculum that then we would implement once the students arrive. And once they got here, we first taught them, started to introduce them to what an archives is and what it's made up of. We taught them how to find things in the archives. I put together like a little treasure hunt, a little scavenger hunt for them to go and look for materials in the archives. We also had resources on how to synthesize their knowledge from primary sources. I had little worksheets with prompting questions to guide their thinking and their writing. Additionally, we visited another archives at the San Francisco Public Library to see how they maintain their archival information. And in the end, the student work was combined into a research article that is focused on historically marginalized academy scientists for publication. And this publication has been accepted into the Bulletin of the Society of Systematic Biologists, and it's in press. Additionally, the digital engagement team at the Academy is working on a digital exhibit in order to communicate our findings to the broader public. So let me tell you a bit about how we chose the scientists that we wanted to highlight. We wanted to focus on people who worked here at the Academy who made an impact on the institution. And this kind of fell into two categories. One were people who were from historically marginalized backgrounds who maybe did not get proper credit or compensation for their work. And we wanted to uplift and celebrate their stories. And then there was another story from someone who had that scientific prowess, but who contributed to the marginalization of others, and that side of his story was not told. And we think both kinds of stories are important to tell the full and true history of the Academy and science in general, in addition to acknowledging our institutional role in racism and misogyny. And once we narrowed that list of 36 down to 7 candidates that were more feasible for us to take on in the timeline and the resources we had, the interns actually were able to choose which candidate that they wanted to investigate further. And as I've heard from them, it was good because then they could share some identity with those people. And we just wanted to pick people who are deceased now. So the people that we investigated span throughout the 170-year history of the Academy. We got to learn about people like Pearl Sonata, who worked in the ichthyology or fish collections for nearly 30 years, but she was also incarcerated at a Japanese internment camp. Inez Mejia was a Mexican-American botanist who began her academic career in her 50s. These stories were reflections of the greater history of the world and academia, and we believed deserved the light of day and wanted to share with you all. In the end, we produced several biographies that you can read now on the Academy's library website. The students also received training from our web development department on actually how to use the content management system and physically upload these biographies to the Academy website, which is a skill that they can take to their future careers. And they worked with our digital engagement team to communicate their work through Instagram stories. So I think it's important for you to hear in their own words why this project was so impactful for our interns. So I'm gonna play a little video for you.
She is the most intellectual driven and just passionate person. She loves what she does.
He spent a lot of his life teaching future scientists.
She discovered about 500 new species of spider.
He treated animals the same way he treated people, and that he was just a kind person.
Oftentimes, who's highlighted in the history of science tend to be white men because those were the ones that were accepted into the world of science. The Academy of History sort of runs parallel to that.
We knew that we wanted to do a project that made use of the archives here at the Academy. The careers in science interns were always part of the plan. I really wanted to not only get their ideas on the project, but I just loved to hear what was important to them, because I think a lot of the things in their own lives, like social justice and making room for more people in science, was what we were exactly trying to do with the project.
They came with a very extensive natural curiosity. They're very excited about finding new information and finding different correspondences and different diaries and stuff that give you a bit more insight on these people that had a massive impact on the Academy of Sciences.
I profiled Gordon Chan, who was a marine biologist. He spent a lot of his life preserving coral reefs, tide pools, and kind of documenting different specimens. He was not only like a leading expert in his field, but he also was able to use his passion to inspire other people to pursue science.
I profiled Katherine Brandegee. She was a botanist at the California Academy of Science and actually the first female curator in the Academy. She reshaped the entire botany department into what it is today, which is this organized and super diverse collection.
I profiled this ornithologist named Luis Felipe Baptista.
He studied white-crowned sparrows primarily and their songs.
All over the city, he could tell the different voices of those sparrows between those districts. They had like each a different song.
Okay, that's typical of this region. Now, this dialect goes south several kilometers to Daly City, which is the next city over. Then in Pacifica, another dialect.
I specifically picked him because he had this whole thing about his like really deep interest and love for languages and like songs, and then he incorporated that with birds. And like I feel like I kind of related to that like interest in like languages because like coming from the Philippines, there's like it's an island or it's a nation full of like dialects. There's just so many hidden figures that we don't know about, and I think it's important to like really bring them out and be like, they are the people who helped us, especially if they're minority, underprivileged people. It's really inspiring for a person like myself.
The library archives hold a lot of like stories that we have yet to uncover.
In natural history museums, which have had a history of racism and colonialism, the folks here have really put a lot of thought and attention into how we can reckon with the past and make good on the future and do better. And I think that one of the things is recognizing where we've come from and recognizing where we still need to go. It's just been long overdue that these people get their credit. All right. Thank you for watching the video. And I'm now going to tell you one of the stories that resonates most with me as a female arachnologist. Harriet Exline Frizzell was an expert on North American spiders, particularly the family Agelenidae. She was one of the first women in the United States to get a PhD focused on arachnology. And she was the first woman to earn the prestigious Sterling Fellowship at Yale, where she worked with Alexander Petrankovich, who was a well-known arachnologist at the time. And together they collaborated on a seminal text in arachnology, the Catalog of American Spiders, in 1939. As for her connection with the Cal Academy, she was a fellow and a research associate here. She actually worked from her home in Missouri, where she was essentially the acting curator of arachnology. The folks here on site in San Francisco sent her spiders to curate from her home. It's clear from her letters and her papers that she was very meticulous, and she really accomplished a lot of work. Among our archives were many letters from around the world, remembering her and showing her impact on the field of arachnology, as well as the Academy, and how deeply she was respected by arachnologists all around the world. Robert C. Miller, who was the executive director of the Cal Academy, once called her brilliant, energetic, effective, and buoyant. She was very kind to beginners, and it's very clear in her letters, too, that she was and very upbeat too. She also liked her semi-feral cats and going fishing. So even though Harriet was clearly valued by the arachnology community and did a lot of significant work for the field and the Academy, she was never compensated for the work that she did monetarily. Ed Ross, who was the chair of entomology at the time, wrote to her once in a letter, "As ever, we greatly appreciate your help and hope that the interest in the material will reward you for your efforts," which means, I hope it's interesting because we're not paying you. And in the same letter, he writes, We will employ a good man to take care of the routine curating that I will become free of. So a man is getting paid to do the exact same work as her. Harriet writes back, please don't worry about my reward for effort on any collection of spiders. Separating such material is hard work, but it is also a great pleasure. Anyway, as a research associate in arachnology for the Academy, I have to earn my title. Additionally, in her own obituary, she maintained that her identity as a scientist was secondary to that of being a wife and a homemaker. And this was in line with the expectations of women in the 1930s to the 1960s when she was actively working. Society told her that her greatest value was as a wife and a homemaker, so it was harder to view herself as a scientist. We infer that she may have had to downplay her own scientific contributions to escape friction from male colleagues and even be allowed to do the work that she was passionate about. So while it was clear she was respected by her peers for her science, she clearly felt that she had to minimize her scientist identity in addition to not being compensated for her work. After her death, Harriet's extremely well-curated collections went to the Cal Academy, where they're still used today by researchers, including my lab. Her husband, Don Frizzell, left $100,000 to the Academy in her memory, called the Don L. and Harriet Exline Frizzell Fund for Arachnological Research. And as a stipulation of that award, Harriet's portrait still hangs in the office of the arachnology curator. And the fund still supports research at the institution today. So while we celebrate her legacy, we also recognize that it took 47 years after her death for the Academy to welcome its first paid woman arachnology curator, who is the current curator of arachnology, Dr. Lauren Esposito, who you all met. So this shows that we still have some room to improve regarding gender equity in arachnology. And now I'm going to shift to giving you part of the reason why these inequities were uncovered and tell you about a person who actually contributed to them. David Starr Jordan is one influential early member of the Cal Academy that we don't necessarily always talk about the full picture of his story. He was president of the Academy and the curator of ichthyology around the turn of the century. And we know a lot about him because he was prolific in his writings and well documented in our archives, and especially at Stanford University as well, where he was also the president. We wanted to tell this story because while his scientific prowess was well recognized, his activities as a eugenicist are less known and equally as important to discuss. So Jordan did have a big impact on the science done at the Academy. He described many, many fish species, including the one on this postcard that was put out by the Steinhardt Aquarium. He also contributed at least 1,900 type specimens to the ichthyology collections here. He also undoubtedly had a big impact on the academic culture and the mentorship of people who went on to work here at the Academy. He mentored people like Barton Warren Everman, who eventually served as the executive director of the Academy for nearly 20 years. In addition to other Academy curators like ichthyologist Rosa Smith and Carl H. Eigenman and Frank Mace McFarland. So clearly he had a big impact on what people were prioritizing in their research and how research was being done here at the Academy. And in between his presidency periods, he kept up giving lectures here at the Academy, and some of them were on scientific topics, of course, but others were on topics like eugenics, including one called The Blood of the Nation. Outside of the Academy, Jordan also actively was holding several positions of power associated with his promotion of eugenics. He was the president of the Eugenics Committee at the American Breeders Association, one of the vice presidents of the first International Eugenics Congress in London, and he served on the inaugural board of trustees at the Human Betterment Foundation in San Francisco, which is an organization that supported Nazi eugenic sterilization. And so he also had influence on some known eugenicists. His other students, in addition to those Academy curators, his other students were people like Paul Popenoe, who was associated with the Human Betterment Foundation, as well as Leo Stanley, who served as the medical director at San Quentin Prison, where he performed at least 600 documented forced sterilizations. Some forced and I think some voluntary. So I think it's important to, like I said, to talk about these things in order to give you a full example of someone who has achieved a lot scientifically, but has also held some pretty egregious views, and who has contributed to the marginalization of the other people in this project whose stories we wanted to lift up. In the end, we had some pretty great student outcomes from our archival research project. The students were able to gain skills in researching, in looking at those primary source documents, and working together to do so, and then synthesizing their thoughts into fully fleshed out biographies. They got to have skills in editing websites. They published their biographies on the Academy website as well as in a scientific journal article. And they were also paid, which I think is really important to note, and important that that's a mechanism that's already built into the Careers in Science program, especially as we reflect on stories like Harriet's. For me, I also had the wonderful outcome, and I hope Kareem did as well, as we got to collaboratively mentor these students and work together to build the curriculum that they went through. And then we learned about the legacies of racism and colonialism at the Cal Academy, academia, and museums in general that we're still dealing with today. And these are perpetuated by individuals like David Starr Jordan, who we met, as well as the systems that him and other people were all a part of. There are some steps to making peace with the past, and we see uncovering the stories that I've talked about today as part of that, and of course, learning from them. In the process of that, we learned that the most unacknowledged people actually just don't have any record or much record of them in the archives at all. So there's probably people who made some significant contributions that we'll never know about because they weren't documented. Additionally, recognizing people and truly giving them credit for their work means paying them and giving them the credit that they deserve as they're doing it. And then finally, that historical figures can be nuanced. I hope that was clear in the story about David Starr Jordan, but it also came up in some of the stories of people that we wanted to celebrate too. Some of the people whose work went uncredited or unknown also held some problematic views that are also important to recognize and realize that they're not perfect people as we celebrate their work as well. All right. Now we're going to pivot from archival research to collections research. And I'm going to talk to you about the molecular phylogeny of the spider family Dictinidae that I built. So this is a pretty different project. So you might be asking, what is a dictinid spider? And the answer is that we don't really know. And that is what we're trying to figure out in this project. We do know that this spider family was first described in 1871 by Pickard Cambridge, and they got their name as the mesh weavers because most of them build mesh-like webs that they use for prey capture that are often found close to the ground in plants. So this is pretty different from the Halloween orb web structure that you may be familiar with. They're generally small and brown. Some of them get to the size of like 2 to 5 millimeters, which is pretty tiny. They're also part of the marinoid clade, which up until recently was another like taxonomically disorganized group that they belong to. Also, other generally small and brown spiders. There have been just a few revisionary studies of the family, and so there's not a ton of information about these spiders, and we wanted to find out more. So as you can see in this map that I made from the data available on the Global Biodiversity Information Facility, dictinids can be found all around the world. So this map includes some specimens and observations from places that were not previously documented to have dictinids. Our study confirms that dictinids are found in Madagascar. The genus Lathes specifically was found for the first time in Africa. And previously there were only 2 known species in Australia. And now we probably have some more. And there's probably more spiders in more places that we just don't even know about yet. So I want to introduce you to some of the incredible diversity within the family Dictinidae. There's a lot of variation, especially in habitat and ecologies. Some spiders, like Amblina borealis, can be found as far north as the Arctic tundra in Greenland. Others in the family, like Saltonia and Certa, are found in salt flats, including in Death Valley, where they sustain some of the hottest ground and air temperatures on Earth. And others, like the diving bell spider, are found underwater. They live their lives underwater and make an air bubble to sustain their livelihoods in ponds. One of the other really cool things about having diverse ecologies is that it's also sometimes reflected in the morphology or the structures that make up the spiders, including the cerebellum. The cerebellum is a plate that produces a different kind of silk called cribbolate silk, which allows spiders, like I said, to expand into diverse ecologies. The cerebellum structure varies considerably within the family, and the family does contain both cribbolate and ecribbolate spiders. The cerebellum has long been considered an important transition in the evolution of spiders, hence why we want to pay attention to it. Letnin's 1967 study was the first to establish that there aren't necessarily just cribbolate and ecribbolate spider groups, but they can be contained in the same family. So to account for all this diversity in the family, it's grown from a single genus when it was first described back in 1871 to 53 genera today. The family is considered a taxonomic Taylor's drawer or junk drawer, where taxonomists have just dumped little brown spiders that they didn't know where else to put. Hence, the monophily and the relationships between the genera remain unclear and is the focus of our study. There have been some attempts to understand what's going on with this family and how it should be categorized, especially in relation to other spider families. Letnin's 1967 study, again, was one of the first examples of phylogenetic thinking, which means like how the family is related to other spider families. And he allied them with other marinoid families, which we maintain today. His study, like I mentioned before, was a bit controversial because he was the first person to say that a family can have both cribbolate and ecribbolate species, and they aren't separate clades. So he kind of caused a bit of a stir with that. Some of the more recent phylogenetic studies also have some disagreement about what species should be included in the family Dictinidae. And I'm going to highlight 2 of them that came out of some of the people from our lab. The Cruz et al. 2019 study was previously the most representative phylogeny of the family, and it included Sanger loci. So this was a phylogenetic study that resulted in the family being deemed polyphyletic. Gournaud et al. Added in genomic data. He had a bit of a lower coverage in terms of the genera represented, but really strengthened the backbone and confirmed the polyphyly of the family. So 2 of these studies also found weak support for a water-associated clade within the family Dictinidae, which is something that we want to investigate further. So as you might imagine, most spiders are terrestrial, or meaning that they live on land. But these studies showed that association with an aquatic habitat has shown up multiple times within the spider tree of life, including in the family Dictinidae. And to be aquatic means kind of a range of things. There are some fully aquatic spiders like the diving bell spider that we talked about earlier that lives its whole life underwater. But then there's some semi-aquatic species as well that can live in the intertidal or salt flats or just generally sandy or salty ground that is close to water. So we wanted to know if the ecologies associated with water arose once within the family or multiple times. And that is one of our aims. So the 4 aims of this project were to reconstruct a phylogenomic hypothesis for the family Dictinidae using ultra-conserved elements. And then using that phylogeny, we wanted to establish the monophyly of the family and really decide which spiders should be in this family. We wanted to evaluate evidence for an aquatic clade and assess the monophyly and relationships among some of the larger and more widespread dictinid genera like Lathes, Amblina, and Dictina. And we face some unique challenges with these spiders. The first is that a lot of them are very, very small, as I mentioned. So for me, this was challenging. Pulling off their tiny little legs in order to extract their DNA was challenging for me, and it was also challenging for the process of extracting DNA because that means there's less DNA available to be concentrated and amplified. Many of the collections that we had access to are actually not fully ID'd to the resolution we would prefer because there's also just not a ton of people in the world who know how to identify these spiders. From the time that this grant that funded this project was awarded, we had a global pandemic in the middle of that. So that interrupted the work, especially in regard to getting specimens from collaborators around the world. And like I mentioned before, we just had limited information to start our investigation. There really wasn't much to go from just those couple of revisionary studies. So the methods we took on to start answering our questions involved sampling, lab work, and computational analyses. We had to gather our spiders and then extract them and build libraries, and then put them through a really interesting computational pipeline in order to gather the data that we could then use to build our phylogeny. And so I'm going to tell you about that novel approach that we have. And it involves using ultra-conserved elements. So ultra-conserved elements are short fragments of DNA that, as the name suggests, are highly conserved among many taxa. And this allows them to act as anchors for aligning them to one another across taxonomic groups. And then on the ends of these fragments of DNA, they're more variable. So then that can be used to account for differentiating different species or different genera, whatever level of that you're looking at. And UCEs are the current gold standard for phylogenomic analysis, so we know we wanted to employ them. However, gathering UCE information can be difficult because they require large volumes of high quality DNA, which is challenging to obtain. So traditional methods use baits in the lab to mechanically isolate UCEs from the genome for sequencing. But because we weren't confident that we could get the depth of coverage that we would need to do that, we decided to take a different approach. So we sequenced a low coverage of the whole genome of these spiders, which allowed us to use the older specimens with poorer quality DNA and because they're tiny specimens with little tissue. We then were able to computationally extract the UCEs using the same probe sequences that you would use to build baits that you would use in the lab. But this just was one more computational step and was a really cool thing that is pretty new that my lab mates and I have been putting together. So in order to make this happen, we first had to start with gathering the spiders. So like I said, we were able to collaborate with a lot of people around the world to send us spiders and also make use of our extensive collections here at the Academy. But there were some spiders that we just didn't have yet. So my lab mates and I took a trip to the southeastern US last year to look for spiders native to that area. We went to several sites in Georgia, Alabama, and Mississippi. And we collected spiders there. And this was really exciting for me because it was a whole new landscape to me. I'd never been to this part of the United States, and it was my first time doing spider collecting. We used a few different methods to collect spiders, which included hand collecting, which is just when you walk around and you see if you can find a spider on the ground and pick it up. We also use beat sheeting, which is when you take like a white canvas and then a stick and you hit a tree or a bush and you see what falls out of that tree. And then you look at your sheet, and if there's something you want, you take a device called a pooter, and it's like a big straw, and you suck it up through your mouth, and then you put it in a vial of alcohol. So I learned how to do that, which was great. We also collected leaf litter, which is where we suspected that many of the spiders we wanted were found, because they like to live in the damp, dark areas of the forest. So then when we got back to our hotel, we would do some work to actually separate and identify the spiders. It's nice to have some experts on our team who know how to do that really well. And then for the leaf litter, we passively separated the spiders and other arthropods from the leaf litter using something called a Berlai's light trap. This is a device that you can see here in which there's a screen that you put the leaf litter on, and there's a hot light bulb at the top. This is not an energy efficient light bulb. It's heating up, and the spiders that don't like to be in the heat because they live in the leaf litter crawl down into a waiting tube of alcohol, where then we take them home. So once we had our spiders, we could start in on the cool extraction and computational pipeline for analysis. So this involved using a specialized extraction protocol, and then library prepping our samples. Like I said, it was a challenge due to some degradation and low DNA concentrations, but we were able to get DNA and later library preps and later data out of samples as old as 1997, which is pretty old for this kind of procedure. And then we sent them off for low coverage whole genome sequencing. And then here comes the really cool computational steps. So once we got the samples back, we were able to clean the raw reads in FAST-P and assemble the genomes in spades. At this step, we also added in genomes from my lab mate Jacob's work, as well as the publicly available sequence read archive on GenBank. And then after converting the files to a 2-bit format that the program Filucci could read, we used existing probes specific to the RTA clade, which is a clade that the spiders belong to, in order to pull the ultra-conserved elements, or the UCEs, from our low-coverage whole genome sequences. Then those UCEs were aligned and mapped, and the data were used to infer our phylogeny under maximum likelihood criteria in IQ-tree. And so I think this is a great opportunity to tell you about two of the huge benefits to this low coverage whole genome approach. One is which you can extract additional information from the low coverage whole genome. There's nothing stopping you from just doing UCEs. You can extract loci like traditionally legacy Sanger loci, like CO1 or 16S. And then you can combine those data with the existing UCE loci in order to strengthen your data set, and you can combine them with other existing legacy Sanger data from GenBank to get more taxonomic coverage and add terminals to your phylogeny. Additionally, having the low coverage whole genome sequences has the benefit of being able to change up the probes that you use to extract your UCEs. So previously, we had been working with a set of spider-specific probes that Kulkarni et al. Had put together. But then in the time of this project, a new probe set that's specific to an even more specific set of spiders by Zhang et al. Came out, and we were able to just switch out those different probe sets with no problem and get a much higher yield of UCEs by several hundred UCEs. So in all, we produced the most representative phylogeny of the family Dictinidae to date. We were able to get 68% coverage of genera within the family, and the previous most representative study had about 38% of the genera. And we were also able to recover large numbers of UCEs, up to almost 2,900 UCEs in one sample, which, if you're not familiar, that's a lot. So I'm going to go back to our aims and just show you specifically how we accomplished each one. Starting first with reconstructing a phylogenomic hypothesis for the family Dictinidae using UCEs. So this is our tree. And before you get too overwhelmed, I know you can't see much, but the important thing here to note is that the backbone of this phylogeny is completely resolved, which is something that has never been previously able to accomplish in phylogenies. There's actually only 5 places on this tree represented by those little pink squares where the bootstrap support, which is like our confidence in how true this phylogeny is. There's only 5 places where that confidence is less than 95%. And the places where these occur are in relatively terminal position, and one of them is even within a species. So we were able to capture a lot of diversity within this family. And when we add in the Sanger loci, we were able to add a few terminals, but the topology was largely the same. So this is our phylogenomic hypothesis for the family Dictinidae. And we can use that big phylogeny that we put together to then determine which spiders actually should be considered dictinids. So this is a simplified version of that tree, and the groups that you can see highlighted are all ones that are considered dictinids at this time. And as such, the family is polyphyletic with Funny Valentine, which is one of the species that's considered to be in the family Dictinidae. It falls outside of the main clade, and so does Bromella. So we determined that those species probably should not be considered dictinids anymore. And I'm going to go into the other clades a little bit more. So like I said, we wanted to investigate the aquatic clade further and see what adding more taxa and genomic data could tell us about this group that had been discovered before, but it didn't have it fully fleshed out. So we think it's possible that it could be its own family. One of the biggest pieces of information that might tell us this is that it's pretty diverged from the rest of the clades in the family that you can see here. Additionally, there's a lot of unique ecologies going on inside this clade, which is, of course, that they're aquatic. And we were able to trace the ancestry of these aquatic traits using an ancestral state character reconstruction. And you can see that the fully aquatic state arose once, and a semi-aquatic state arose a few times within the family. And we think that the ancestor of the clade was likely terrestrial. And combining this information with information about the corbellum, that really cool characteristic that we talked about earlier, we can see that there is some activity going on evolutionarily in relation to both the aquatic organisms as well as the evolution of the corbellum. So there may be some correlation between the morphology of the corbellum as well as being associated with an aquatic habitat. But this is an area that needs some more research. This is just a hunch that there might be something going on. And because we found some evidence of evolutionary changes within this clade, we wanted to run a Bayesian analysis of macroevolutionary mixtures in order to see if we could detect any rate shifts within our phylogeny. And if we did, they would have showed up in red on this tree, but no rate shifts were detected, which is a bit surprising given the interesting changing ecologies of these spiders in the aquatic clade. And finally, we wanted to determine whether some of the larger and more widespread genera within the dictinid family were monophyletic and what their relationships were like. So starting with Lathes, we think that it's possible that Lathes might also be its own family. This, again, is pretty deeply diverged from the rest of the dictinids. And it's almost monophyletic. There's one other genus within the family called Benayella. But this is an area that needs some further investigation and needs to be compared to the morphological studies as well. But having this information of the genomics is very important for making those future conclusions. And then finally, we really wanted to look at the clade that contains Dictyna and Amblina, because these are also some of the biggest and most widespread genera within the family. And we just have rampant polyphyly in this family. Each line that's a different color represents a different genus. So this is clearly an area that needs future investigation. And when you get to the more terminal spots that mostly just have Dictyna and Amblina, it's really not clear that there's a Dictyna clade and an Amblina clade. They're really just all flip-flopping. This is a mess that is going to be taken on by another person in our lab. So we were able to conclude that we produced one of the most representative known phylogeny of this family to date using genomic data. We recovered 68% of genera represented in our genus, which represents a huge collaboration from people all around the world and no small feat. We were able to confirm that dictynids, as they are now, are polyphyletic, with Funny Valentine and Bromella outside the main dictynid clade. As we evaluated evidence for an aquatic clade, we were able to see that most aquatic dictynids end up in that one clade, but not every aquatic dictynid and not every aquatic spider, of course, by any means. But we were able to identify a clade of closely related spiders, many of which with aquatic habitat associations. And that association is often correlated with changes in the corbellum structure. So that's something that our morphological team wants to look at more. And we were not able to detect any rate shifts, evolutionary rate shifts in this group. We also saw that Laphes, being quite divergent and quite different from the other dictynids, it's possible it might be its own family. I can't conclude that today because we need to add in the morphological information to see if it's in agreement. And that Dictyna and Amblina are not reciprocally monophyletic. There's several other genera in the clade. It's generally a mess that needs to be addressed with further work. So to end today, I want to tell you a little bit about what I learned and where I'm heading next. So as I talked about in the beginning of my talk, I've been using collections in new ways. I hope that I've demonstrated to you today how much untapped information is held in museum collections, just waiting for the right project to come along and make use of it. I've learned that our stories have power and deserve to be told, whether that be of people who are uncompensated or uncredited for their work, or just little brown spiders that people didn't really care that much about before. Spider systematics will continue to benefit from the advent of new technologies like UCEs and genomic data that I've employed in my study, but we need to keep up the morphology-based methods as well. My work is only beginning to understand the relationships using genomic data, and future work in the lab will take this information and use it as a framework for interpreting the morphology that we see in the spiders. I also learned that I just really like doing systematics and biodiversity science research, and this is my first time working with arthropods, and I hope I get to keep working with them in the future. And so I didn't get to talk about all of the activities that I've done over the last 2 years, but I'm really grateful that I've gotten experience in collecting lab work, computational work, writing, mentorship. I've contributed in a few ways to our collections here at the Academy in spiders and scorpions. I will, specific to my master's research, I will have first authored 2 papers and co-authored another paper about the projects that I told you about today. I've also presented 2 of those projects at academic conferences. I will have mentored 14 students and given 8 other sort of less formal scientific talks, as well as 2 posters. So I'm pretty proud of all of this. And looking ahead, I want to write up my findings that I told you about in my spider project to be in a manuscript for publication. My lab is continuing to research gender trends in spider species descriptions, looking at who is writing species descriptions of spiders and where there might be some inequities there. This also comes in part to acknowledge Harriet's work. Well, I hope to stick around the Academy for another year and continue to wrap up my projects and mentor students. I'm going to plan to apply for PhDs in evolutionary biology this coming fall, and I hope to start in the fall of 2024. I want to keep working in natural history collections and biodiversity science while making an impact on my field and the public. And overall, I want to stay curious. And I want to thank all of the people who have contributed to the production of this project and my entire master's. Of course, my current lab, as well as past members of the lab and past people who've worked on the family Dictinidae, the marinoids in general. Thank you to Athena and Lynn in the lab for guiding me through that. I want to give a special thank you to Rebecca Kim for offering me so much support throughout my first year and in the archival project. And then other folks who have worked at the Academy who have just really supported me also emotionally and with mentorship, like Dr. Rebecca Johnson and Maya Jones. Thank you very much to my funders, like the NSF for funding my spider project, the Cal Academy for hosting me, as well as, of course, San Francisco State University. Thank you to the Arcs Foundation for funding a lot of my livelihood as well, and my tuition. And I want to thank all the people who sent in specimens that would not have made this project possible without collaborators from all around the world. And then I also just want to end with a couple more special shout outs, especially to folks in my lab. Thank you to Franklin Calariquelme for thinking so many things that I do are funny, and for being a morphology king. Thank you to Sarah Cruz for everything you say being funny, and for teaching me how to make my first phylogenies 4 years ago. Thank you to Jacob for being my ride or die lab bestie, my desk mate, my therapist, and one of my best friends. Thank you to Lauren Esposito for believing in me, giving me this opportunity, and for being an inspiration to me every day in science. I'm so grateful for everyone who has supported me in my time here at the Academy and at San Francisco State. All of you on the slide and many others. I just couldn't fit that many pictures. Thank you for being here. Thank you for all that you've done to get me to this place. It means so much to me that I've been able to grow so much as a scientist and a person during my time in my master's. And so thank you very much. And I'll end and see if you all have any questions.
Okay, first off, just amazing job. Such a great talk. Also, congratulations on that tree. It looks incredible how it came out so well supported. My question was on your Bayesian analysis of your rate variation. And I was wondering if you think there might be, the reason you might not see any variation is because of using UCD as something that's very conserved. And if you think there might be a different result if you looked at whole genome or different genes. Yeah,
thank you for that question, Ezra. I think that that's something I hadn't considered before, and I think it would be cool to see if we use different data, how that might change. The tree that we used was the UCE only tree to build that BAM analysis. I didn't run it actually on the tree with the Sanger data as well, so that could potentially be another way. We want to try to run it again too with maybe messing with the settings a little bit. We just use the default settings that came with the program. So we want to do a little bit more investigation on other studies that have done, have used it for spiders specifically and see if we get anything else. So yeah.
Great job, Kate. That was awesome. I was wondering if you have an idea of what you want to do for your PhD, if you want to continue with spiders or what you want to do.
Great. I'm going to repeat the question just in case, I don't know if people online can hear it, but it's okay? Okay. Well, for my PhD, that's a great question. I'm down to continue with spiders. Like Lauren said, I'm kind of down for a lot of different things. I just think that it's really cool to work in the field of biodiversity science and specifically at a natural history museum. And so I want to keep doing that. And I'm open to my study organisms changing. But like I said, I really like arthropods because I think that there's so much that we don't know about them and so much diversity that we haven't yet recognized. So I would love to study them, love to stay in systematics, and also maybe explore some other questions too. So I hope that answers your question.
Great talk, Kate. So Lauren touched a little bit on some other projects that weren't necessarily related to your master's, but were some research projects that you've been involved in. Would you like to talk about those?
Sure. So I've kind of moonlighted and also worked on a few other projects. I'm wrapping up my undergraduate honors thesis on frogs and newts. I just got the proofs back the other day. And so I'm working on that, finishing that publication. And then I also worked on a nudibranch project that was part of my SSI project, my summer internship project that will also hopefully eventually be published. Yeah, I think those are the two main ones that aren't explicitly part of my master's, but I got to do. Also, Lauren talked about the seminar that I helped run about racism and colonialism at the Academy. That was kind of something extra I did just because I care about those issues. Thank you.
I think there's a question online. If someone joining virtually asks, is the spider named Bunny Valentine described? And if so, where does it live?
It is described, and it lives in China.
Great job. I'm curious if you have some future directions for the archival research and if you want to incorporate that kind of research in your PhD or future endeavors.
Sure. I don't personally have any set plans to continue with the archival research. I know that project is awesome, and it's actually been picked up and continued on in the Careers in Science program. Marie Angel, who works here, and Rebecca Kim are continuing with that project. And I've had the privilege of actually listening in on the Careers in Science students at their final wrap-up session, and they've been continuing to look at some more people, and I'm excited to see what they do.
Just a follow-up question about the archive project, because I was curious if at any point in time there were you could reach out to any of the descendants of the people that were part of the projects and what that might have been like.
Yeah, that's a great question. And we did do that to a degree. Some of the photos that were in that video actually came from some of the descendants. I actually also met one of them in the Gap once. I was shopping and I was just talking about my project. She was like, wait, I think that's like my husband's great uncle is Luis Felipe Baptista. And so I got to talk to her, which was really cool and unexpected.
Great job, Kate. I have a question also about the archival research, and I wonder, when you all were doing the research, if you could think a little bit about what we should be doing now to make sure that people's stories are told, not only just now, but going forward. Did you learn anything from?
That's a great question. I think just as you're doing your projects, making sure that you really try to think of everyone who was involved, which might be actually expanded as what we previously thought deserved credit. So with every project comes lots of stakeholders. There's people who own the land you work on, who've traditionally stewarded that land throughout the past. and who just do all kinds of other things that contribute to the project that might deserve either authorship or at least acknowledgement in the acknowledgement section. So I think that's something that me and my lab especially have been thinking a lot about. And additionally, of course, one of the big things was paying those people too when they were able to contribute, and also just thinking about how we can keep expanding that realm of people who are involved and are given credit and are given proper compensation. Does that answer your question? Thank you.
I have a question.
Yeah.
So it seems like you developed a pretty massive skill set over the course of 2 years. And I'm curious what was the most challenging skill to develop and apply?
The most challenging skill to develop, I think technical skills were like learning how to do a lot of the computational stuff because that was pretty new to me. But also in general, I had to learn skills about just like navigating like social interactions in academia as well, which can be challenging, and just how to be a more professional person and more accommodating and inclusive. And I think that doesn't always come easily, but I think is one of the most important things to grow as a scientist, and one of my goals also to grow as a person throughout this master's.
All right. Let's give Kate a round of applause.